Hi everyone, I'm Jaya Vishwanathan and you've found the No Brainer podcast where I discuss all things neuroscience in delightful conversations with various experts. I hope you enjoy today's episode.
Hello, everyone, and welcome to another episode of the No Brainer Podcast, Season 2. And I'm very, very excited to have you all join us today because I have two guests, and they're both experts in their fields. And I'm very excited to both introduce you all to them and also have this conversation with them. And I'm really eager to learn from both of them. So, first, let me introduce you to Dr. Ann Cohen. Dr. Cohen is an associate professor of psychiatry at the University of Pittsburgh School of Medicine. She's also the director of the neuroimaging core of the University of Pittsburgh Alzheimer's Disease Research Center and the co-director of the molecular biomarkers and psychiatry program. Dr. Cohen is also the lead of the recruitment team on the Alzheimer's Disease Biomarker Consortium Down Syndrome, ABCDS, and is the PI of multiple NIA-funded studies focusing on neuroimaging in aging and Alzheimer's disease. Dr. Cohen earned her PhD in neuroscience from the University of Pittsburgh in 2006 and completed a postdoctoral fellowship at Western Psychiatric Institute and Clinic at UPMC in 2011. Dr. Cohen served as a steering committee member for the neuroimaging professional interest area of the Alzheimer's Association International Society to Advance Alzheimer's Research and Treatment, or ISTART, from 2015 to 2019, and is a member of the ISTART Advisory Council. Her primary research interests focus on PET imaging to explore early detection of amyloid in Alzheimer's disease using the amyloid imaging agent Pittsburgh Compound B, and the relationship of social determinants of health to Alzheimer's disease pathology. Dr. Cohen is co-leading the partnership core of the ACE study. She's also the mom of two fantastic kids. So welcome, Dr. Cohen, and I'm really excited to have you here. Next, I would like to introduce you all to Dr. Blair Braden. Dr. Blair Braden is a behavioral neuroscientist with expertise in neuroimaging and neuropsychology. Her core roles at Arizona State University are Associate Professor in the College of Health Solutions, Program Director for the Speech and Hearing Science, and Director of the Autism and Brain Aging Laboratory. Her research encompasses a multidisciplinary program studying aging in autistic adults, from basic science to translational treatment, factoring in the largely unexplored area of sex differences. They are among the first to comprehensively describe age-related cognitive and brain changes in autistic adults, and two, to develop behaviorally and neurobiologically informed interventions for autistic adults across the lifespan. They focus mostly on cognitive compensation, mindfulness-based, and social strategy techniques. So welcome, Dr. Braden. I'm very, very excited to have you both here. And before we get started with our discussions, I also want to just say, you guys have really vast expertise in multiple topics, but today's conversation is going to be focused on your research, specifically in autism in older adults and in dementia autism interaction. And so you guys are the leaders in autism research, which is a very important and yet understudied area and misunderstood area. And so because especially there is so much misinformation about autism in the public, I'd like to start at the very beginning. And if you can explain autism and neuroatypical development conditions for our guests, I think that would be a really wonderful starting point. Thank you.
Okay. Yes, I can dive in first.
Please.
So I think it's important for people to understand that autism is an exclusively behaviorally defined condition. So although, we talk about neurodevelopment, which means brain development and different pathways of brain development, autism is not defined by anything objective about the brain. It's defined by symptom presentation. So there's two types of symptoms that someone needs to show to receive an autism diagnosis. The first is social communication challenges, and the second is the presence of restricted and repetitive behaviors and interests. which is a really wide net, that captures diverse things from repetitive motor movements, all the way up to very specific focal interest in a certain topic.
Yeah, I think that's a great starting point. And I think the other thing that is really important that Dr. Braden just mentioned was this idea that we're not diagnosing a scan, we're not diagnosing a blood test. I am by training an aging and Alzheimer's disease researcher who's much later to this area than Dr. Braden is. And one of the, I think, real challenges that the aging field is having with some of these things is that we rely very, very, very heavily on biomarkers. And the Alzheimer's disease field in particular is in this whole new era of biomarker diagnosis. And when we're talking about autism and autistic individuals, you're not talking about a scan or a blood test. You're talking about a specific sort of phenotype. And so merging those two fields together and getting researchers and medical professionals and the world to speak the same language is really, really challenging, because I think a lot of people think about autism as something like hypertension, which you do a test and you know that it's there. And that's not how autism is defined. I think it is one of the reasons it's ripe for this kind of misunderstanding about autism.
Yeah, absolutely.
Yeah, so you have already raised some really interesting points because, I think both dementia, right, and not specifically Alzheimer's dementia, but just dementia in general can have multi-etiology causes. And then because of just what you've told me about autism, I think because it's based on the symptoms and the symptoms can maybe have many different causes, you're trying to combine your research from two very heterogeneous spaces. So it's fascinating that you're doing that. So I would love to learn more about what your individual research questions and interests are, and also, what you enjoy about working in this space.
So I guess I can start this time.
Absolutely.
So, as I said, I'm newer to the study of autism in the context of aging, but what brought me to this interest was really this idea of social determinants of health and the idea that your brain doesn't age in a vacuum. Aging is essentially the process from development all the way through senescence. And so when we think about the aging brain, the context really matters. And as I began to learn more about different communities and the way different lived experiences, the way different environments impacted the way these individuals aged, and additionally came to study Down syndrome and aging, one of the things that became really important to me was this idea that if a brain starts out different, it's going to age differently. You can't assume that every brain ages along the same trajectory. And the thing that puzzled me and bothered me about that is that neurodevelopmental differences like autism, like ADHD, like dyslexia are often exclusions for studies of cognitive aging and Alzheimer's disease. So we have a whole population of people who are being left out of those studies. And particularly these biomarker frameworks that we've developed in Alzheimer's disease. So this idea that Alzheimer's disease is caused by first the emergence of the amyloid beta protein, followed by emergence of the tau protein, followed by neurodegeneration, and then cognitive decline. And what the field has found more and more as we expand our inclusion into different populations is that this is not a one-size-fits-all framework. And so that's really where I became incredibly interested in this and began to seek out the expertise of folks like Dr. Braden, who have been studying this for a very long time in a lot of contexts, with this idea that we can really start to put our heads together and approach this in a large and systematic way to get answers for these populations, who many of whom are aging into the age of risk as we speak.
Yeah, so I can kind of pick up that that's what we set out to do almost 12 years ago, was to be one of the first studies that compared aging and middle-aged and older autistic folks to people who aren't autistic, that we try to make, as similar to our autistic people in every way, shape, and form, except that they're not autistic. So we can draw conclusions that if we see different aging trajectories, then it's probably something about aging with autism that is creating that different trajectory. Aging research takes a long time because you have to wait for people to get older to see the differences. So we've started to see some differences. But it's also hard to find middle-aged and older autistic folks because the autism diagnosis didn't even exist when they were kids.
I see.
So they, most of them have made their way to understanding that they're autistic in adulthood. So, some people were identified in younger ages, even though it wasn't really a DSM diagnosis back then, but the understanding existed. So anyways, we've always been a small study. Our findings have always been very preliminary. And also, back when we started this study, the biomarker framework in Alzheimer's and dementia didn't even exist. So now we're at a place where we're ready to scale up and do this approach multi-site all across the country, where we can find lots more autistic adults in this age range and evaluate this biomarker framework in this population.
And another thing that I want to just jump off of there that I think is really important is something that I'm really interested in, and I think is something that we need to consider, especially as we consider trying to be as inclusive and diverse in our ability to study folks as possible, is that I think we're going to have to be clever about how we identify autistic individuals and give the opportunity for people who, for a variety of reasons, may not have received a diagnosis, whether that's older adults where the diagnosis didn't exist, or that's women who still unfortunately are less likely to be diagnosed, or people of color who are less likely to be diagnosed. So I think that's another area. While the study that Dr. Braden just mentioned isn't going to focus on that, I think this is a really important thing that the field needs to be thinking about, is that while a diagnosis is a gold standard, often because of a variety of factors, people get left behind in those diagnoses. And thinking about that, I think, is going to be a really important piece of this puzzle as well.
Yeah, no, I think you have raised some really great points. And I think there's maybe some fear of getting like an autism diagnosis and like social stigma associated with both like dementia, although like the dementia stigma is certainly on the decline in a lot of cultures, but there's still, I think, so much stigma around going to a psychiatrist or any mental health provider with an issue. So it's great that you guys are already ahead of time, like thinking of these issues.
Yeah. And, being able to be as inclusive as we can in the stage of not requiring that someone have a previous diagnosis to participate in the study, but being able to do our own diagnostic evaluations in our studies is a huge part. And then also, we, as you mentioned in my bio, have a really strong focus on the inclusion of women. So we will always go for including 50% women in our studies. And we get really close, which is saying a lot when the diagnosis ratio is so much stacked the other direction towards males. But yeah, we find that autistic women are very passionate about participating in this type of research and bringing awareness to autism in women.
And to give you the pat on the back that you won't give yourself, there's not many cohorts that have been able to achieve that with women. I mean, it was a huge lift, and that you all have done it is pretty incredible.
That's a great segue, because I kind of want to pull at a couple of things that you've talked about. So can you talk about your cohort a little bit more? And then specifically, like, I think both of you have talked about aging trajectories as well as biomarkers. So specifically, like, what kind of aging trajectories are you looking at comparing autistic versus non-autistic adults? And then when you bring in dementia, and then what kind of biomarkers are you thinking of or working on?
Do you want to take the trajectory and I'll take the biomarker?
Yeah, so our cohort is open to adults of any age, so 18 to as old as we can find them. Once people hit 40, or if they come in over the age of 40, then we follow them longitudinally. So that means every 2 years we ask them to come back and participate again. And what participation looks like is doing a couple hours of cognitive assessments and doing an MRI scan where we look at the structure of the brain, but also how the brain works using functional MRI. So our study, 18 and up, we're getting pretty close to about 400 people.
Wow.
Of those 400, about 200 are, maybe 250 are over the age of 50, and we try to get them to come back every 2 years. Not everyone does, but we do our best. But some people that started with us in the beginning that are still hanging with us, we've followed them for 12 years now. And you can really start to see how things change for people when you follow them for that long. So some of the things we're looking at for change is how cognitive abilities change. And when we say cognitive abilities, that's a big, wide term. So some of the things we're most interested in are memory, for one, because memory is going to be the cognitive domain that declines the most in most people as we get older. And then executive function, too, is really important to us because that's the domain that autistic people are more likely to struggle with from even a young age. So wanting to see if those early challenges in executive functioning make someone susceptible to then those becoming harder and harder as they grow older. And then, of course, there's things we know about the brain as to, what parts of the brain support these different types of cognitive functions. So we want to see if those brain structures are changing differently. Are they getting smaller, faster, for example? And then also the networks that are communicating with each other to support these functions. Does that communication change differently as autistic people grow older?
And in terms of the biomarkers of interest, for this study, at least, so we actually just enrolled. Dr. Braden got to meet him because he popped into someone's office to say hello today. We just enrolled our first autistic participant in our preliminary biomarker study, which is measuring Alzheimer's disease biomarkers and neuroinflammation biomarkers. And so these are the two things that for me that I'm most interested in. I think there's also, after I talk about these biomarkers, maybe Dr. Braden can talk a little bit about some of the Parkinson's disease markers that they're looking at. But we're doing both blood measures and brain imaging measures of the presence of the amyloid beta protein in the brain. So this is the protein that makes up the plaques in Alzheimer's disease. So we're doing Pittsburgh Compound B PET, which was essentially the first amyloid imaging agent that allowed you to see amyloid in the living brain of individuals. And then we're also doing a tau PET scan. So tau is a protein that becomes over-phosphorylated. So these phosphate groups get attached to tau. During the Alzheimer's disease process, it's thought to be triggered by beta amyloid. And when tau becomes hyperphosphorylated, it sticks together and it makes those tangles that live inside the nerve cells of the brain. And so we use PET imaging to measure both of those. And then we also use PET imaging to measure the inflammatory response in the brain. And then we can compare where the Alzheimer's pathology is in the brain relative to inflammation. I think in particular, the inflammation question is very important and interesting in autism because we know that there's a variety of health risk factors and health comorbidities that autistic people may have due to a whole variety of factors that are likely to play a large role. Just the stress of the lived experience of existing in the world with autism has a long-term impact. And so being able to measure that in the brain, we hope will be a really important first step to understanding if the sorts of cognitive changes that Dr. Braden was talking about from her cohort, if those are a function of more Alzheimer's disease pathology in these individuals' brains, or is it more nonspecific? So are we just looking at changes that reduce cognition because of more inflammation in the brain, which may lead to more loss of brain cells? Right. So the more information we can collect in terms of the biomarkers and follow these individuals long term, the more likely we will be to understand the mechanisms behind those changes that are being observed with cognition and structurally in the brain.
Okay, so that's great. And I love that you guys have complementary approaches that's gonna, and hopefully I think, and I believe in this, that the whole is gonna be greater than the sum of its parts. And just by the fact that you're collaborating, hopefully the insights will be more than double. But you talked about comorbidities and autistic specific comorbidities versus comorbidities that, might be dementia related or lifestyle related. Like we've talked on this podcast earlier with neuroimmunologists who talk about like some immune factors that can bias you one way or another. But I also know that you have a specific interest on sex differences. So do you have any insights to share already about some of the sex differences that you are seeing in your data, what that means going forward, as well as some of, because anyway, I kind of think of it as like there's some things you can prevent just by like lifestyle changes or exercise and nutrition and things like that. And then there's things that might need a pharmacological intervention, things like that. So kind of like just what are you seeing so far from your data sets and your research, both separately and together, to talk about the comorbidities and sex differences?
So one thing that I can touch on that kind of gets at both of these things that we haven't directly mentioned yet is genetics. So when Annie's talking about neuroinflammation as being a particular risk factor in autism, I think the lived experience of the stress is part of that, but also a lot of research suggests that autism risk genes are related to the immune system in the body.
Oh, I did not know that.
So,
okay, that's
so
fascinating.
Autistic people can be genetically predisposed to have a different inflammatory experience and have lived stress that
Right.
could really, be a double hit. So, and that's just one area of genetics. So then also building on genetics, we've genotyped our participants in our study, and the strongest sex differences we see so far, and keep in mind, my study is still relatively small for making conclusions about aging. And then if you split it in half across the sexes, it's even smaller.
Yeah.
But we see that our autistic men are carrying more of the Alzheimer's risk genes than our autistic women. And then to even understand, why is there a sex difference in autism diagnoses, as genetics is also a possible answer to that as well.
Right.
So I think that there's so much unknown about autism in the two sexes and aging differences in the two sexes. And I think genetics will be a part of answering the differences that we see.
Yeah, I think that's such a great point. I don't understand genetics very well, so I tend to ignore it, which thankfully for Dr. Braden, she keeps me honest about it.
Okay.
I think another area that is really critical, and I hope your listeners pick up on that, we're saying we think a lot, because this field is really, I mean, 12 years sounds like a really long time, but Dr. Braden is, early in her career. She has many, many more years, as I hope I do as well. And
Absolutely.
12 years is not a long time for this kind of study. And so, all of these areas that we talk about, there's so much more work to be done. But I think the mental health aspects that come along with autistic adults, and so, for example, late life depression is a huge risk factor for the development of Alzheimer's disease. We also know that women are more likely than men to develop late life depression. We also know that autistic individuals are more likely to have depression, and it's also more likely to be refractory for like general typical treatments for depression, and there's a higher rate of suicidality. And all of these pieces, as you start to put them together, do they add up to, the question becomes, do they add up to an increased risk of just a profile of a brain that's under stress? Do they add up to, you have this two hit of genetics plus all of these stressors? Is it the environment that individuals live in? So you have on average, lower SES, less likelihood to get quality medical care. All of those pieces put you at higher risk for Alzheimer's disease.
Yeah.
So the question really becomes, is this just sort of a terrible a terrible pot of risk factors that all come together?
Yeah.
Or is it genetics and the risk factors? I think the data is really bearing out that it's going to be a combination, but disentangling these questions is going to take decades, in my opinion.
Absolutely. And I think good science often takes long because you're adding to the collective knowledge bit by bit. But one more question I have about this topic is also, and again, I'm not an expert, that's my disclaimer, but people talk about like high functioning autism versus non-high functioning autism. And so from what you've told me, your cohort sounds to be all inclusive and, encouraging participation from everyone, which is fantastic. But is that a myth? Is there biological bases to like classify people as such? Acknowledging that this is very preliminary and you're working on lots of different aspects of these data. And if there are publications that you would like to share, please feel free to do so, and I will make sure to include them in the show notes for you guys tuning in. Yeah, so is this idea of like high functioning versus low functioning or whatever the terminology, is that a myth?
Yeah, that is a very timely discussion that is being had on a lot of levels. So first, let me say that our study has not been inclusive of people with what we would say are higher support needs. So that is something, as Dr. Cohen and I move into hopefully a much larger funded effort, that is top on the list of priorities, is to include autistic adults with higher support needs who have not been included in this research in the past, my study included.
Okay.
And there's, reasons for that cognitive assessment looks very different in people with high support needs. And I have never had the great collaborators or the resources to be able to tackle that on my own, but we will tackle that as the effort grows and moves on. And I'm kind of answering part of your question that high functioning, low functioning are terms that are generally thought of in the past, and people talk more about the support needs of someone, that someone is either high support needs or low support needs. And then woven into this is intellectual disability. So about a third of autistic people have intellectual disability, and generally those people are who you would consider as higher support needs. But it's not an absolute. There are certainly people who are autistic with average or above average IQ who also have high support needs.
Okay.
It's just more likely to be if they have intellectual disability.
Okay.
And I'll add to that this challenge that Dr. Braden talked about, that is related to what cognitive testing looks like in individuals who have intellectual disability, is not unique to autism. This is something that, in my experience with studying Down syndrome and Alzheimer's disease, which is a very different population because they have a very clear-cut genetic risk for Alzheimer's disease, that typically these studies have excluded individuals who are more severely impaired or who have more severe intellectual disability. It's very, very difficult to do cognitive assessments when individuals don't have average language skills. It's very difficult when individuals are nonverbal to assess cognition because so many of the cognitive tests rely on that. And so the ability to measure cognition and those capabilities in individuals with higher resource needs and potentially lower IQ or lower language abilities requires a lot of technology and a lot of expertise that is different from what we're used to doing in cognitive assessment. So thinking about things like the way the eyes move, those are things that are actively being explored, but it's been a real problem in understanding cognition and intellectual disability. How do you meet people where they are and measure their cognitive capabilities?
Yeah, I mean, that's a great point. Thanks for the clarification as well. And I think that's very important to know. But I think it's really great with these longitudinal cohorts because you have a person's own baseline to then look at their individual trajectories as opposed to like trying to link a group average. I'm very optimistic about the insights that your combined research efforts will have. now and going forward.
Yeah. And if it's okay, I'd love to make a point on what you just said about longitudinal, because I think that's something that is very nuanced about aging research, that there's kind of two ways to do it. There's following the same people over time, which is longitudinal and is really the gold standard because you're comparing someone to who they were before. But the faster way to do it is to recruit a bunch of people across a broad age range and look at correlations between age and performance and say, older people are performing worse than younger people. That must mean aging affects this cognitive measure, right? Absolutely. And one of the problems in the aging world is what we call cohort effects, which means that all the different life experiences someone who's 60 years old today had versus someone who's 20 years old today. And in the world of autism, you could not get larger cohort effects between a 60-year-old autistic person and a 20-year-old autistic person.
I see.
So it's really, there is a fair amount of literature out there that looks at age correlations in autism. And we don't think it's necessarily going to be representative of what the research is going to show when we continue to move forward with longitudinal.
Yeah, I think you couldn't have said it better. This is so important. If you think about just the way, and again, not to bring it back to Down syndrome, but we've talked about this a lot in Down syndrome as well. We're having the first generation of people who entered midlife with autism, intellectual disabilities, autism with intellectual disabilities, Down syndrome, who were not institutionalized, who were included in education with supports. That we know that has a huge effect on the brain and the way the brain ages and the capacity for people to achieve certain things. So as Dr. Braden said, it could not be more important to understand how these schools age as compared to themselves. But it's expensive and it takes a long time, and that's not real satisfying for the world.
Hey everyone, hope you enjoyed this episode of the No Brainer podcast and see you at the next one.